Hi, my name is Connor Gallimore, and I'm a graduate student in Dr. Jordan Hamm's lab here at Georgia State University. Welcome to the Hamm Lab virtual tour. Today, I'm going to be talking about some of the tools and skill sets that we use to answer questions about how sensory cortices of brains process contextual influences. Being a graduate student in basic and translational sciences offers us many opportunities to learn a diverse array of skill sets that often extend beyond our undergraduate training, such as soldering electrical wires, for instance. This engineering technique allows us to forge electrical contacts, building useful circuits for things like automation or electrophysiology. Here, I'm repairing contacts on a printed circuit board that allows us to record EEG-like signals from the scalps of mice. Another technique we commonly use is confocal microscopy, which allows us to investigate anatomical structure and connectivity within and between brain regions. Now you may be wondering, why are we using some of these tools and skill sets, and what kind of questions does the Hamm Lab seek to answer? Now, if you remember previously, I mentioned that our lab is interested in contextual processing. We participate in all sorts of different contexts throughout our lives, in our household, amongst our social groups, our work, our school. However, researchers have devised a simple way to assess context by asking, does something fit within a defined pattern, or does it deviate from that pattern? And we call these sensory oddball paradigms. There are auditory oddball paradigms, in which a tone can be repeated over and over and thus made redundant, and interspersed at some point in between these redundant tones will be a deviant tone, a pitch-shifted up or down tone. Visual oddball paradigms look very similar, except for they use a visual stimulus, such as an oriented grating, and the deviant stimulus would be applying some sort of rotational shift to this grating. Now, in EEG studies of humans, it's been shown that this standard stimulus, this repeated one, will register in a way that is actually very different from this deviant stimulus. And we call that different response cortical mismatch negativity. Interestingly, certain neuropsychiatric diseases cause deficits in this registration of a deviant stimulus, such as schizophrenia or bipolar disorder. Let's take a closer look at what happens at the microcircuit level. Shown here is one of the staple techniques in our lab called two-photon microscopy that allows us to record activity of visual cortical neurons in an awake mouse in real time as they're watching visual stimulus. Now, when we use one of these sensory oddball paradigms, let's see what happens. You can see that with redundant stimulus presentations, the activity of these neurons dampens. However, when a deviant stimulus comes up, the activity increases, and there's a sustained, augmented neural response. Research, much like what I've shown you here, can provide a better framework by which we can understand the results from human studies. In neuroscience, there are many levels of analysis that we can look at. We can look at very small scales at the level of genes and molecules, or very large scales at the level of entire brain networks, behavior, or our interpersonal interactions. Conditions such as Alzheimer's or Parkinson's disease may result from cellular dysfunctions or molecular interactions gone awry. Other psychological conditions may result from deficits at the behavioral or interpersonal levels. We think that schizophrenia may result from abnormalities at the circuit or ensemble level, local levels of sensory processing, or even ways in which brain networks, sensory cortices, communicate with each other. Though I hope this simple example makes it easy to see how context shapes many aspects of our lives, the way we recognize patterns, what we notice about the world, and it's important to consider as we navigate. I want to give special thanks to my colleagues, Jacob Holmes for his 3D print work, and Dr. Jordan Ross for her neural recordings. Other members of the Hamm Lab are listed here as well. And of course I have to give song credits for the classical piece I played before. I hope you enjoyed this virtual lab tour.